नमस्कार। ई कंटेंट मॉड्यूल में आपका स्वागत है। Students, today we are going to discuss on the module of bacterial reproduction, transformation, transduction and conjugation. In this module of part 1 transformation, we will be discussing the chapter under General Microbiology paper of Biotechnology in BSc Semester 1 of Institute for Excellence in Higher Education. Dear friends, in this discussion, we will be discussing some points about the transformation. After studying this lesson, you will be able to define transformation in bacteria, describe the process of transformation in bacteria. Explain the importance of transformation. Students, you have already studied in this course Basic and History of Microbiology, Evolution of Microbiology, Classification and Microbial Phylogeny, Microbial Diversity, Maintenance and Preservation of Microorganisms, Microbial Growth, and Microbial Metabolism. But the importance of microbial diversity comes through this topic of transformation. That is, in this topic, if you say that the cause of bacterial diversity is transformation, transduction, and conjugation. And in this session, we will be discussing about the first topic, that is the transformation. Bacterial reproduction is a fascinating process in which the transfer of genetic material between bacterial cells takes place. Bacterial reproduction includes transfer of genetic material between bacterial cells, allowing them to adapt and develop, and allowing them to thrive in diverse environments. So you can see here that bacterial reproduction is a process in which the genetic material of the surrounding is transferred inside the bacteria, allowing them to adapt and develop. Now come to the transformation. Transformation is a process by which bacteria take free DNA from their surroundings and incorporate into their genome. This phenomenon was first observed by Frederick Griffith in 1928 during his study on Streptococcus pneumoniae and Diplococcus pneumoniae. He found that if non-lethal bacteria and the heat-killed lethal bacteria are suspended together, the non-lethal bacteria can become lethal when exposed to heat-killed lethal bacteria, which is evidenced by the transfer of genetic material. A suspension containing a mixture of heat-killed toxic encapsulated cells and living non-toxic non-encapsulated cells was injected into the mouse. A small fraction of the living bacteria turned into the viral encapsulated type. The transformation capacity was inherited by the non-viral bacteria, which is converted to newly transformed living strain. So, in this way, Griffith found that there is something which is transferred from heat-killed bacteria to the non-lethal bacteria. So from the heat-killed bacteria, something comes in the suspension in the surrounding of non-lethal bacteria, and then it enters inside the non-lethal bacteria and convert it into the lethal bacteria. This is the process of transformation where the non-lethal bacteria is transformed by this genetic material into the lethal one. E. coli generally does not pick up foreign DNA, but it can do so in the presence of calcium chloride. This experiment is known for the identification of DNA. Now come to the process of transformation. When the recipient cells of the bacterium are grown in the presence of killed donor cells, the change is observed. Here you can see the cell of the bacteria having the circular DNA as plasmid, and normal irregular bacterial DNA. The first process is the bacterium become competent to take the foreign DNA. This process is known as competence. Change is possible only when bacterium cells are able to take extranuclear DNA fragments and incorporating them into their genome. These extracellular DNA fragments are incorporated in the genome. Calcium ions help neutralize the negative charge of the DNA molecule, making it easier for them to pass through the bacterial cell membrane during the transformation. So bacterial cell membrane become permeable. The development of the competent state is related to the cell density. Thus, most of the cells that grow in culture become competent when a significant number of cells are obtained. Therefore, capacity represents the transient stage of the life of the population. The time and duration of its occurrence are characteristics for a bacterial genus. The ability can be induced by a variety of factors, including environmental cues and a specific genetic mechanism. Not all bacterium species are naturally competent, but most of be induced to become competent. This competency is due to the DNA binding protein, which is also a competence factor. External factors trigger the expression of genes involved in this activity. These genes encode proteins that facilitate DNA uptake and recombination. During the capacity development, bacteria produce a variety of proteins responsible for DNA uptake. These include DNA binding protein, transport system for the DNA, and enzymes involved in the processing of incoming DNA. This factor acts by altering cell surface properties either by the formation of receptor sites or by increased permeability of the donor cell. Then cyclic AMP was also found to be important in playing a role in capacity development as well. When it is added to the medium, this compound greatly increases the level of capacity between cells. Now come to the regeneration and incorporation. Now the foreign DNA, or it is called as the transforming DNA, come in contact with the this bacterium near to the DNA binding proteins. Once a bacterial cell is able, it is more likely to pick a free DNA fragment from its surrounding. These DNA fragments can originate from other bacterial cells that have come into the environment through lysis, or that is the cell background. Double standard donor DNA molecules bind to receptor sites on the surface of the receptor cell. Donor fragments are brought to the surface of the recipient cell by endonuclease in a shape that varies in different bacterial species. After attachment to the recipient cell wall, donor DNA is actively transported inside the cell immediately after regeneration. One strand of the donor DNA fragment is degraded so that it becomes single stranded.